All right, so today we will continue our discussion on PCA, and then we will introduce briefly this technique called GLM-PCA, standing for Generalized Linear Model PCA. So for this, I would say to thoroughly understand it, you will need to have some background on generalized linear models. But since this is not something we have covered yet, I will try to give a high-level idea about what it is. And then MDS standing for multi-dimensional scaling, which has some very strong connection to PCA, but its input is different. So for MDS, which I will introduce at the end of this lecture, or maybe the beginning of next lecture, you no longer provide an observation by feature matrix. Instead, you just provide a similarity or dissimilarity matrix of your n observations. So it's like pairwise symmetric matrix. That's your only input. And then MDS is trying to find some two-dimensional or three-dimensional embeddings of your observations so that in this embedding, in this visualization, because you can visualize 2D or 3D, the pairwise distances preserves the input pairwise dissimilarity. That's the goal of MDS. So it's a visualization technique, but it has strong resemblance to PCA, and it's also a linear technique. So first of all, we're still here, right? After these steps, we are performing linear dimension reduction. A very brief review of what we covered so far. Seurat object, right? I said you input a gene by cell count matrix, transform it into a Seurat object. And the first pre-processing step is to filter out those bad quality cells. That's the first step. So you will remove columns, not removing genes, not removing rows. And then after that, you use normalization to remove the effect of cell library size. So there are multiple ways to do that. One is to set every cell's library size to a constant, like 10K. And then you do +1, then log. So the idea here is to make the data more like Gaussian distribution. And also you hope to stabilize the variance. That's some perspective we asked in your first homework. By stabilizing the variance, essentially, we want to make every gene's variance unrelated to its mean. So this normalization is basically data transformation. You transform your data. And again, it's not monotone transformation, right? So after the normalization, a gene's relative expressions in different cells, the rank order may be changed. They are no longer the original rank order based on counts. And then this step is to select genes. So if this filtering step is to select cells, then this highly variable feature step is to select genes. So we don't use all the genes, which could be as many as 20,000 for human genes, but we're only keeping a few genes that have the top variance compared to its mean. So that's the default method in Seurat, the VST method, which actually compares every gene's variance to its mean by fitting a curve, right? And only the genes whose variances are much bigger than expected variance given the mean are selected. And this highly variable gene number is by default in Seurat set to 2,000. But this number, I would say it's arbitrary. Why 2,000? Why not 1,000? Why not 3,000? We don't have a very solid justification as to my knowledge. But I think it's just some number people think are reasonable, 10% of all genes. But this number is also under the assumption that, oh, you believe the large variances capture biological variation, while the genes with small variances, the majority of them only capture technical variation. That is assumption. So then we have the data with the dimension as 2,000 rows, the selected genes, and the remaining cells. Is there a question? That's a good question. I would say in practice, some people may filter out the genes that are expressing less than 5% of all cells. Yeah, but I would say in this object, zero object, we analyze in the tutorial, maybe that step is already done. Or I would say probably you can also filter out those genes in this highly variable gene step. Because if a gene has too many zeros, too few non-zeros, the variance wouldn't be large. Yeah.
If we're normalizing the data to remove the effect of mean on variance, then how can we fit a curve with mean and variance afterwards?
That's a very good question. That was the question I had about the default option for highly variable gene selection in SURE. That's the VST method. If you recall in that lecture, you can check the slides. What I said is that interestingly, in the VST method, the log mean, log variance are based on count data. Yeah, so that's something I found very interesting because they're not using normalized data from the previous step, but they go back to count data. Yeah. And that's a very good question. And then with those selected genes and remaining cells, you do scaling. And here the scaling is done per gene. For each gene, you consider to be one variable, which is one row. You center it to have mean 0, and you scale it so its standard deviation becomes 1. That's what you do for every gene. And then we come to PCA. So PCA is after all these, right? But the scaling, just to clarify, it's done on the normalized data. So the hope is that after all these steps, every gene becomes approximately standard normal distributed. Okay, normal 0, 1. Okay, and this is just the simple illustration we used in our last lecture, right? What is each PC? Each PC is a linear combination of original features, which are genes in your data. And we consider each PC to be a mega feature. It's a combined feature. And we hope that it's more informative. So then the key question is, it's informative in what way? All right, so here is just the result of PCA, what it looks like. So basically, if we only have 2 original features, then we can have at most 2 PCs. So if you have 2 PCs, then you don't lose any of your information in your data. You're basically doing just rotation and scaling to define PCs. So essentially, I would say, maybe I should take back. I should say the PC, each PC is a linear combination of the original features because the linear combination, those weights are coefficients in the unit vector, right? Their length is 1. Then it's a rotation. Basically, you can see PC1 is feature 1, feature 2 combined in this way. PC2 is feature 1, feature 2 combined this way. And we define the 2 PCs to be orthogonal to each other. Right? And in our last lecture, we said that then you will work in this new PC coordinate system, not the original system. So you have PC1 and PC2 as new features, basically. So I want to just pause here a little bit to ask you, is there any difficulty in understanding this connection to our single cell data analysis? Because in single cell data analysis, each dot would be 1 cell, okay? And each original feature would be 1 gene. That's the connection. But just a few days ago, I had a conversation with a journal editor who is interested in single cell data analysis, but has no background in statistics or computation. So I found it very intriguing that the biggest question the journalist had was, why do we say a cell is in the high dimensional space? What does high dimensional space mean? I don't know if any of you here can give an answer. Why do we say we start from high dimensional space and we use PCA to reduce dimension? Then what does the high dimensional space represent? Yes.
All different genes.
Yes, other different gene. Any other answer?
Oh yeah, I was just gonna say like just the lots of genes or intelligence to like analysis.
Yeah. So basically, why do we say a gene is a dimension, right? If we have 2,000 genes, then you have a 2,000 dimensional space. Every cell is a point in that space, right? Why do we say that? The reason is because in every dimension defined as a gene, every cell has a value. So then every cell has one value for each gene in each dimension. So every cell has 2,000 values. That is the cell's coordinate in that 2,000 dimensional space. So I guess the challenges here is probably that you can never visualize a 2,000 dimensional space. That's probably where the challenge is. But we can only visualize up to 3 dimensions. That's just our human mind. But we just imagine something like the 3D space. Anyway, thank you for the answers. So now this is something I found super interesting. So I want to share this with you. So this is from this Stack Exchange website about PCA. So this one, I strongly suggest every one of you to read it. There's a link here, making sense of PCA, eigenvectors, eigenvalues. So give a very intuitive understanding or intuitive explanation of PCA, not using math, but using the intuition. But I think the quote in this thread to be very interesting. So it says, you do not really understand something unless you can explain it to your grandmother, right? This is from Albert Einstein. Then I want to ask you this question. Can you explain this? Why PCA, eigenvectors, eigenvalues? What was the need for these concepts? That's question 1. Second question, how would you explain these to a layman? And I want to share this with you. I think when I was a graduate student, Stack Exchange was a very helpful resource. It has a lot of information about statistics and programming, right? You can find a lot of answers there. Actually, I would say nowadays, if you think about what ChatGPT provides to you, it's a summary of internet content, but it does the automatic summary. Previously, when you do Google search, you do your own summary, right? But I think the summarization process is learning process because you have to digest the information and come up with your answer. But now ChatGPT makes things faster. It just digests information and provide to you, but it's not guaranteed to be 100% accurate. So therefore, I would say ChatGPT is something useful if you are an expert, right? For example, if you are a writing expert, you can take the output and revise it. It might be easier than writing on your own. But if you, for example, if you don't know English well, then probably it's not something very good for you because then you would just trust whatever it gives you. So same thing here. So I would still think it's very important to do the Google search and find the information yourself, right? And also, even one of the 3, pioneers of deep learning, I believe it's Yann LeCun, he had made a very specific point on, I saw his slides on Twitter, on X. He said that ChatGPT is not supposed to be used as a search engine. That's not the intended use. Rather, it's just a language tool. You can consider it to be something to smooth out your language, to help you fix the grammar for those kind of purposes, but not as a search engine to provide information because there could be some bias, right? Because it cannot go beyond what's out there on the internet. It's a summary statistic. For example, if many people are saying the same thing, even though it's a biased information, but it will be summarizing that way. Anyway, so any of you want to give this a try? First question, why PCA? Why do we need eigenvectors, eigenvalues? After last lecture, any of you can explain it to your grandmother? I think it's very good that you first have this question, then you read this thread to see how the original post, the author explained it. Yes. Sure, thank you.
Yes.
Yeah.
And it's very hard for like the human mind to like see how genres relate to each other. And so PCA provides a way to condense all of these different information categories that you describe objects into a smaller number of dimensions that we can actually visualize. And I guess if you want to get more mathematical about that, there's obviously that eigenvector, eigenvalue. essentially provide a solution to the problem of like how do you sort of project your the things you're trying to describe into a lower dimension space. And that was something that was confusing to me initially. And after I kind of studied it, like it made more sense to me that I feel like eigenvectors, eigenvalues are almost kind of a very convenient solution to a very problem that seems kind of complicated. And it's a very nice way that kind of breaks down that is something that like you know that's
Yeah, that's a very good answer. Yeah, it's very good answer. So it's the right high-level summary of what PCA aims to do. So in this post, the author used the example of wines. So say you have many wines, and every wine can be described in multiple characteristics like the color, the taste, etc. Then how would you summarize or compare your wines, for example, in one dimension? So you can have one score per wine. How would you do that? Then you can use PC1. You can use the first PC as the way to rank your wine, right? So then the PC1 is the way you summarize the multiple characteristics of your wine into one number. And then that number can hopefully be used to describe the quality of your wine. So we can rate our wine, say, okay, this is a better wine than the other. So if you just use one PC, ranking becomes possible, right? But if it's multidimensional, it's not easy. Yeah. So I would say in many real scenarios, when we give something a score, we're trying to do a summary. And we hope the score can be correlated, positively correlated with many important features, like the wine's color, taste. And also, so how to say eigenvector and eigenvalues. So for this, yes, you can say they are concepts from linear algebra, but I really want to raise your attention or draw your attention to linear algebra, because I would say for data science, if you want to analyze data, most of the data are in a matrix format. So I would say for data science applications, I would argue that linear algebra is more useful than calculus. Even though in college, maybe calculus is still paid more attention to, and the curriculum, the textbook is more standard. But linear algebra, the teaching is very tricky because you can teach in a very technical way, just all of the calculation, or you can teach it in a very intuitive way. So that's why I strongly recommend that MIT open course if you want to learn more about linear algebra, because that will just give you more capacity if you want to understand more about the methods. So that's why I think it's really worth learning. But other than that, coming back to the topic, eigenvalues, eigenvectors, we can think about it in the way of geometry. So say that you have original features, some of which are highly correlated, right? So in this case, if you start with the covariance matrix, so in the covariance matrix, the dimension is feature number by feature number, and it's a symmetric matrix. In the diagonal, you have features variances. The off diagonals are pairwise covariances between two features. That's what the covariance matrix is, right? Covariance, mathematically, it just means that how these two features co-vary together. When one's variance is big, is the other variance also big? That's the intuition behind covariance. But given that covariance matrix may have some off-diagonal entries, what this eigenvector, eigenvalue thing is trying to say that I want to combine my features in a way so that the combined features become uncorrelated, and the original features total variance. So basically, how much variance is explained by the combined feature, that's the eigenvalue, and how new feature, how each combined feature is combined from the original features, that's given by eigenvector. So basically consider eigen decomposition just as a way to do the rotation of your original coordinate system in the way that, so for example, your original two features, coming back to this example, they are indeed positively correlated, but we want to find a new coordinate system so that the two new features are orthogonal. So that's just the way we think about eigenvalue and eigenvector. So essentially here, the two weights, 0.866, 0.5, that's eigenvector 1. Of this original two features will give me a 2x2 covariance matrix, and 0.866, 0.5 is the eigenvector 1, negative 0.5, 0.866 is eigenvector 2. And then the eigenvalue is the length of the two vectors. I would say not the length of the two vectors, but basically you project the data to the two new dimensions, then the variance of PC1 is eigenvalue 1, variance of PC2 is eigenvalue 2. So we give you the details of the derivation in our last lecture. But today, we hope to give you more intuitive understanding. So yeah, I think this is a very good blog post because it has different levels of explanation from the simplest, highest level to the most detailed one. So I suggest the reading. So back to the question we got at the beginning, PCA has two goals. So the goal is to find new features as linear combinations of the original features, and we call the new features PCs. So the new features should satisfy two goals. The first one is that we want to maximize the variance of data. So the goal is to have PC1 explain most of the variance, PC2 explain most of the remaining variance after PC1, PC3 explain most of the variance after PC1 and PC2, etc. So each PC explains the most of the remaining variance. And we showed that by maximizing the variance, the derivation will give us an eigen decomposition problem. So mathematically, it becomes an eigen decomposition of the covariance matrix. That's what we said last time. But just be careful that when we see the covariance, we actually have each feature centered at 0. So that's why X transpose X. So X is N, let's say here we use the traditional notation, X N by P, N observations, P features. So here, when we say X transpose X is P by P, right? This matrix is the covariance matrix of the P features if each of the P features are center 0 in X. That's the key thing. If it's not centered, then X transpose X is not a covariance matrix. I just want to clarify that. So we talked about the detail last time for goal 1, but goal 2 is what we will continue talking about. Allow reconstructing the original features as well as possible. So say that we have P original features, we reduce them to K PCs. Then the K PCs are mathematically proven as the best K mega features that can allow you to reconstruct the P original features. Okay, because there are other ways to come up with K features, but the K PCs are the best one for reconstructing. That is known as a reconstruction theorem, but we will not talk about that here. We'll just show you how the reconstruction is done. So this is a cartoon, again, I found from this website, but a different post, I think by the same author, which I think is a very nice cartoon. So what it says is that, suppose I'm looking for PC1, and PC1 is known, we want it to be a linear combination of the original two features. So you are basically centering PC1 with the same center of feature 1 and feature 2. This is the center. And there are so many ways to define PC1, right? You're just rotating the possible lines for 360 degrees. And then you want to see, okay, for each line, I project original points to the line. So the red dots are the projections of my end data points to PC1. So these are what I call as PC scores. Every observation has a PC score. And PC1's weight for feature 1 and feature 2, that is my loading, right? So you can see that at different lines, the scores are different, and sometimes the red dots are more spread out. So let's see if we, oops, sorry. So if we just right there, right? So the red dots are most spread out. So that's what we call maximize the variance. But if it's right here, you see the red dots are very crowded. The variance is small. So that explains why we think this line, the purple line here, is the PC1, in which the projections, PC1 scores have the largest variance. In the meantime, if we think about reconstruction, then reconstruction means that you will use a one dimension to represent the original two-dimensional data. Then, of course, you want this purple line. This line best represents the shape of your data, right? Intuitively. So that one-dimensional representation is definitely better than this, because this one, if you have a line like this, it's very different from your original data. I don't know if this intuition makes sense to you. So we consider all possible P1s and we stop at that purple one. That's where we have the maximized variance of PC1 scores and also the best reconstruction of the original data. Okay, so no more questions. Then this is, yes. No. In that case, remember, we have this formula given to you. The maximum number of PCs you can find is the minimum of two numbers. One is n-1, one is p. Whichever is smaller, that's the number of PCs you can find. And the reason is n-1 is because of centering. We actually center every column to have, center every feature to have mean 0. In that way, you loses 1 degree of freedom, because in that case, you're basically looking at the n values for each feature, center them to 0. Yeah. So let's just imagine this, right? You can think about this as the extreme example you just said. All the points lie perfectly in a line. That's when the two features are perfectly correlated, right? Perfect dependent. But you can still find PC1, right? Once you have PC1, then PC2 is just orthogonal to PC1. You still have PC2. But here it's just that in this case, all the data points will have 0 scores in PC2. That's just it. Yeah. Okay. So this is just a review of the last goal. To maximize the variance of data, in our last lecture, we found that the loading vector of PC1 is the eigenvector corresponding to the largest eigenvalue of this X transpose X. And then PC1 score is just X right multiplied by the loading vector, right? Because the loading vector is its dimension is P by 1, the P dimensional vector. X as n by P, n observations, P original features, then X multiplied by the loading vector will give me n by 1. That's the NPC1 scores. For the second goal, reconstruction, we will give you more detail today. So this is again from this blog post, which I found to be very useful. So I borrowed the explanation here. So let X raw, let me try to pull out the laser so you can see. Yes, let X raw be the n by P data matrix with n rows, data points, and P columns, variables or features. After subtracting the mean vector mu from each row, so we center the data, we get a center data matrix X. That's what we work with, right? Every column has mean 0. Let V be the P by K matrix of some eigenvectors that we want to use. So just to clarify, the eigenvectors are eigenvectors of X transpose X. So this means that we have K PCs from top 1 to top K. These would most often be the K eigenvectors with the largest eigenvalues. You go through the largest eigenvalue, second largest eigenvalue, etc. Then the N by K matrix of PCA projects so-called PC scores will simply be given by Z as X times V. So V is P by K, X is N by P. So together, Z is N by K. So then we have every data point, or we say observation, with K new dimensions, and that's the K PCs. Okay, so just remember, X is the centered data matrix, Z is the reduced dimension data matrix, projected data matrix. Okay, reconstruction. Yeah, this is a very nice illustration. What it means is that, okay, for PC1, we found it, and then we plot the N data points in PC1. So now this x-axis is PC1. So maybe I'll just add something for your understanding. Where's my cursor? Okay, so what I'm going to say here is that this is feature 1. And this is original feature 2. And this is PC1. So that's why every data point here has a PC1 value, corresponds to the projections here, the red dots, the projections. And then for reconstruction, on the right-hand side, we go back to the original space of feature 1 and feature 2. So essentially, what we are doing is that given the PC1 score, we use the definition of PC1 as a linear combination of F1, F2, feature 1, feature 2. We rotate it back. So then we are restoring the data points in the original space, but only using PC1 information. So you see that the data points are in the original 2-dimensional space, but they are only explained by PC1. So it's a reduction of data. You see the effect is that you reduce the scatter. Yeah, you reduce the scatter or along this line, right? And hopefully, we think this is a noise reduction. So essentially, you believe the PC1, this captures the signal, and you believe the scatter, the vertical, perpendicular, I should say, perpendicular distances are due to noise. If that's your belief, then you can only keep PC1 for your next step instead of using the whole 2-dimensional data. This is the intuition about why PC1 gives the best 1-dimensional reconstruction of your data. Yes. No, there's no PC2. We're only looking at PC1. So PC1 is this new dimension that is defined as some combination of F1 and F2. And then you just project every point perpendicularly to this line. And you record these positions. And these correspond to these values in PC1. That's it. Yeah. So we didn't look at PC2 at all. But PC2 has this orthogonal relationship with PC1. Yeah, that's something we know, but we are not looking at PC2 here. Yeah. Okay, we didn't do scaling here. The scaling, so in this other discussion so far, the data X is only, every feature in X is only centered. but not scale to have standard deviation 1 in the PCA introduction. But it has that in our single cell data analysis. So that's different. But no, I think you confuse two things. One is the unit variance. One is correlation. So what I'm saying is that even F1 and F2 both have variance equal to 1, but if they're perfectly correlated, the points are still in a line. They wouldn't be in the cloud. They wouldn't be in a circle. They're only in a circle if feature 1 and feature 2 are uncorrelated. Right. So the variance and correlation are two things. Okay, so here basically you can say that given the blue dots, given their x-axis values, we can calculate one variance. Given their y-axis values, we can calculate one variance, right? Probably now without the scaling, here the variance in the x-axis may be 2, in the y-axis maybe 2.5. So they may not be equal. But once you do scaling, you make the variances equal. But if they have correlation, the correlation will still be there. Then the correlation, the stronger the correlation, the tighter the points will be close to a line. Yeah. Okay, these are great questions. Then let's look at the technical stuff, reconstruction. So in order to be able to reconstruct the original two variables from this one principal component, we can map it back to P dimensions with V transpose. Just reminder, what is V? V is P by K. So then every column of V is one eigenvector or one loading vector. So then we have the first column for PC loading 1, second column for PC2 loading, and the kth column for PCK loading. So it's a feature by PC matrix. Then V transpose is K by P. So we map the K PCs back to the P original features. Indeed, the values of each PC should be placed on the same vector as was used for projection. Okay, so compare subplots 1 and 3. So what it means, compare this one and this one. Compare these two. Okay, the result is then given by called X hat. As Z, recall Z is N by K. It's your PC score matrix. So Z N by K, P transpose K by P. Then together we get N by P. Same dimensions as the original X, but we call this X hat. So mathematically, as Z is X times V, this is what we have from the previous slide. Then we multiply V transpose on the right end. So this X times V transpose. So recall V is P by K, V transpose K by P. So together we get a P by P matrix. So it's a transformation, it's a projection, right? But you see that what we actually have is originally your X may have P full column rank. So if you remember, column rank and row rank are two linear algebra concepts. If your P columns have this linear independence, that means you cannot explain any column as the linear combination of the other columns. Whenever that holds, then your X has P full column rank. And then after you do this, because V transpose by nature has only a rank of K instead of P, even though it's P by P, but it only has K in K columns in V and K rows in V transpose. So you can check that this square matrix, this is a square symmetric matrix P by P, has only K rank K instead of rank P. And we arbitrarily set K to be smaller than P, right? Otherwise, there's no dimension reduction. K smaller than P. So essentially, when you multiply X by this rank K P by P matrix, you reduce the column rank of this X hat to be K instead of P. So essentially, you can think about X hat to have less dimensionality, less dimensionality than original X, which is reflected here. So even though here, this is X hat, this is X, you can see that they are in the same space. They roughly have the same shape, but this matrix X hat, which is still n by 2, right? 2 features, but it has only rank 1 because the 2 columns can be explained by 1 vector, right? So it's rank 1, but here it's rank 2. You cannot explain these 2 features perfectly by 1 feature, by 1 new feature. But if in the original data, the data points are perfectly lying in the line, then it's essentially rank 1, even though it's in the 2-dimensional space. So just to give you some summary, we can say that a line is 1-dimensional, a point is 0-dimensional, right? And a plane is 2-dimensional. So basically, if we have a line in the 2-dimensional space, then the essential rank is only 1, even though you have a 2-dimensional space. That's what the X hat has. So basically, this X hat as the reconstructed data matrix has only rank K because it's based on K features. Yes.
Like the perfect movie of positive. Everything will be perfect.
Independent, yeah. You will have a bowl. Yeah. It's unidentifiable.
Exactly.
Yeah. 1 dimension. Yes, that's right. Yeah, exactly. The perfect intuition. Exactly. So we use PCA just because features are correlated. Yeah. Yeah. Yeah, because otherwise, if you have like in the one example, right? If these characteristics are totally independent, there's no unique way to combine them into one score. Yeah. Okay, so basically, this is the word explanation of this formula. The PC reconstruction is the PC scores, which is Z. Z is PC scores times eigenvector transpose. That's V transpose. That's what we get for the X hat. But when we say plus mean, we mean that we add the subtracted mean mu back to X hat to get X hat raw. So the reason is here. We first subtract mu from X raw to get X, then we perform PC on X, and then we reconstruct X hat. But these are all based on centered features. So if we want to restore the center back, we add back the mean. Okay, so this is just like this. Let me just give you one more explanation. This is the original data. When we say we do centering, we basically move the center to 0, 0. That's what we have for this data. But before we do the shift, probably the center is 1, 1, right? Then what we do is that after we do the PCA, do the reconstruction, here the center is 0, 0. Then we shift this new data to center 1, 1. Then we move it back. Okay. So note that one can go directly from the first subplot to the third one by multiplying X with a VVT matrix, which is here. It is called a projection matrix. So this VV transpose is called a projection matrix. If all P eigenvectors are used, then PP transpose is the identity matrix, P by P identity matrix. Then no dimensionality reduction is performed. Hence, reconstruction is perfect, right? So if we go from PC1, here we keep both PC1 and PC2, then we have perfect reconstruction, which is exactly here. In this case, when we use this PC1, PC2, we didn't lose any information. We keep the whole data set. And, okay, if only a subset of eigenvectors are used, for example, the top K eigenvectors, then this VV transpose is not an identity matrix. So it's basically, how to say it? It's basically, you can check it. So it's not an identity matrix because you are not using the full information. So then you have some information loss here. Like I said, X transpose is M by P, but it has only K column rank, while X has n by p, but it has p column rank. This works for an arbitrary point z in the PC space. So Z, recall, PC space is k-dimensional, right? So Z is a k-dimensional data point. It can be mapped to the original space by Z times V transpose. Because V transpose is k by p, you project it back to the p-dimensional space. Okay, so what it means is that, say, these are my n data points by PC1. Suppose that I pick another point here in PC1, then I can use this way to go from PC1 to the original space, the rotation back. I can find where the point is in the PC space, but it will also be on this line, right? So for example, if it's in this end, then it will be this point. If it's in this end, then it will be this point. Okay. Okay, so now this one more thing. This is related to our single cell data, because once we do centering and scaling, then X transpose X, P by P, becomes correlation matrix. So the covariance matrix is reduced to correlation matrix because every feature now has variance 1. So when two features have variance 1, then their covariance is just equal to their correlation. This is a very important fact, I think, from probability. So then when PC is done on correlation matrix, our single cell case, then the raw data, which is before centering, before scaling, that's the normalized data we had. It's not only centered by subtracting mu, but also scaled by dividing each column by standard deviation sigma I for I feature. In this case, to reconstruct the original data, one needs to backscale the columns of this X hat with sigma I, and only then to add back the mean vector mu. So you need to scale it back. So for example, if feature 1 originally have standard deviation 3, then during the scaling process, you divide that column after centering, you divide the center column by 3. Then you do PCA, right? And then once you get X hat, you need to multiply this column 1 of X hat by 3 to multiply that back and then shift the center back. So we need to worry about the scaling for reconstruction, not just the centering. Any questions? No. Okay, so more technical detail about PCA. So these are the things I won't cover, but for those of you who are very interested in the technical detail, I suggest reading these two posts. One is that there are 2 ways to do PCA. One is, as we said, eigen decomposition of X transpose X or SVD on X. So they will both give a way to give you the loadings and loadings and eigenvalues, but they are not exactly equivalent. For example, SVD will give you singular values. Eigen decomposition will give you eigenvalues. There's a relationship, but they're not equal values. However, if you want to get the eigenvectors of X transpose X, you can basically get them from the right singular vectors of X. So there are some mathematical equivalents here. So I suggest that you read this post if you're interested. Second interesting thing is that the terminology loadings. So in this class, I think also in most PCA literature, we refer to loadings as the eigenvectors of X transpose X. However, this author has a different opinion. He thinks the loading should be eigenvectors scaled by the square roots of the respective eigenvalues. So just 2 ways to call what is loadings, but he has some reasoning, I would say, why the latter is a better way to represent loadings. But anyway, so what we say about loadings are the eigenvectors, just to avoid any confusion. This is something important to know, because otherwise you may confuse what is the loading here, right? Because just to make sure that there are 2 ways to call loadings, but which way is the one used in one paper, we should be clear. Okay, so this is what we didn't actually finish last time about the single cell PCA benchmarking. Recall that the paper's authors have 4 data sets with very different numbers of cells. from 700 cells. So by numbers, probably. Anyway, so basically 4 large data sets with very different number of cells, very different N, the gene number is almost on the same scale. So I think, yeah, maybe it's just, I think the number is right. Maybe just the position of comma is different because you see the file size is indeed much larger than this one. Anyway, so the author's goal is to, interesting. I'm not sure whether this is right. Now it caught my attention because it seems impossible that for the third row, the file size is, for log CP_med, log CP_med is only 9.3 megabytes, right? Because this one is much bigger than the previous row, but this one is much smaller than the previous row. Yes. Sorry? I think so. Yeah, I think so. That looks right, but it's a typo here for sure. Yeah. Thank you. So for these 4 data sets, they just compare different PCA implementations, algorithms, and see for the 2 large data sets, the standard PCA and this IRLBA PCA, they couldn't run on them. Only the algorithms that does the so-called online learning or incremental learning, so which means that it doesn't eat all the data into the RAM at once, but it does this online computation based on batches, many batches of data, 1 mini batch at a time, and then update the PCA results. Only these algorithms can run on the 2 large data sets, and the downsampling can run. By downsampling, it means that you downsample a subset of your cells, run PCA. But they pointed out that the downsample PCA is plot. So after they do PCA, I should say, they input it into t-SNE. The t-SNE plot will look different in some critical ways after downsampling. So obviously downsampling is not recommended. But this is a useful table showing that once you have this gene multiplied by cell number of genes multiplied by number of cells. Based on this number, you should decide which algorithm to use. So the critical values are 10 to the 8th, 10 to the 10th, right? These are 2 boundaries. So suppose that our gene number are all approximate after the highly variable gene selection. We have the gene number as 10 to the 3rd, right? So then if you divide 10 to the 8th by 10 to the 3rd, you have 10 to the 5th cells. If your cell number is in the range of, in the order of 10 to the 5th, you could use the standard PCA. If your cell number is 10 to the 5th to 10 to the 7th, you could use the IRLBA PCA, which we'll talk next. But if your cell number is larger than 10 to the 7th, which means that more than 10 million cells, right? That's very large, then you should use the online algorithms. Yeah, so that's just a practical suggestion. And for R, Python, Julia, they have their corresponding versions. So what is IRLBA? It's called fast truncated SVD and PCA. The IRLBA function is designed to compute a partial singular value decomposition. It is largely compatible with the usual RSVD function, but there are some differences. In particular, the IRLBA function only computes the number of singular values corresponding to the maximum of desired singular vectors. So you need to specify the K beforehand. So otherwise, the standard PC will compute all PCs, right? From the top 1 PC to the top maximum PC, which is the minimum of n-1 MP. You will compute everything. But this will only compute up to the top K PC. K is what you specify. For example, if K singular vectors are desired, so this is just in the SVD, you specify the number of left singular value, singular vectors, right singular vectors, but that's fine. So in this case, it's just equal to K for our PCA purpose. Then only the 5 corresponding singular values are computed. The standard RSVD function always returns. So this is the standard SVD for standard PCA, always returns the whole set. So it takes longer than this truncated version. Then the IRLBA function is an iterative method that continues until either a tolerance or maximum number of iterations is reached. But just be careful, the SVD or this function, SVD or PRC, PRComp, these two for exact SVD or exact PCA, they are exact. But IRLBA is approximate, it's not exact. Okay, problems with difficult convergence properties are not likely to be encountered, but the method will fail with an error after iteration limit is reached in those cases. So after that many iterations, you still don't have a convergence. Yeah, so this is the IRLBA. And then for the fastest one, most efficient one, out of the core, or it's online learning or incremental learning. So these terms all refer to this strategy. So it's a technique used to learn from data that cannot fit the computer's RAM. So you're not reading the whole data set in. The ability to learn incrementally from a mini batch of instances. So instances in our language, observations or data points, they are equivalent terms. Instances are used mostly in machine learning. So sometimes called online learning is key to out of core learning, as it guarantees that at any given time, there will be only a small amount of instances in the main memory. The other data are in your hard drive, but not in your memory. Choosing a good size for the mini batch that balances relevancy and memory footprint would involve some tuning. So basically, you want to make sure that you use your memory as much as possible, but in the meantime, it's not something too much burden for a computation. Yeah.
If it's basically instance of cells or the cells in
Only cells. Only cells. Yeah.
is this like the implementation of notices of sketching in Seurat?
I'm not sure. I'm not sure if it's sketching in Seurat. That if you look here, let me see. Here, SK, no, it's not listed here. But for sketching, I think it's downsampling. That's my impression. I think it's downsampling rather than online learning, but you can double check, yeah. So downsampling is easy, but it's not the ideal way. All right, so then let's just quickly move to the next part, but we'll finish soon. So this is about how to determine the number of PCs to include, right? Especially if you want to use the IRLBA, you need to specify the K, pre-specify the K. How do you do it? Okay, so this is the key issue. To overcome the extensive technical noise in any single feature for SCRNA-seq data, Seurat cluster cells based on their PCA scores, with each PC essentially representing a meta feature that combines information across a correlated feature set. The top features, therefore, represent a robust compression of the data set. However, how many components should we choose? 10, 20, 100? For this purpose, the elbow plot is the way to go. What it does is that it basically calculates, so this is standard deviation, which is the square root of the eigenvalue. So the square root of eigenvalue PC1, PC2. So we see that top PC explains more, later PC explain less, but we want to look for the elbow point where the curve basically flattens. And that's what we think, okay, including more PCs have only a marginal benefit. So the increased dimension doesn't worth the increase in explaining the variance. Because if we think about this as not a square root, just the lambda, just the eigenvalue, then each eigenvalue is the variance, is the variance explained by each PC. So top one PC explain the most, second PC, second most, etc. So these dots are guaranteed to be monotone decreasing, but you want to look for the elbow point. So for most people, they may just do the visual check and look for the elbow. But next time when we come back, we'll talk about this method called, I'm just here, the needle algorithm, which can give you an automatic way to find where the elbow is. Okay, so we will stop here and we will resume on Wednesday morning.